Hello everyone, welcome to Biology for Knowledge channel. Here I'm going to talk about another chromatography technique that we all come across today and we are all using is known as affinity chromatography. So what do you mean by affinity chromatography? Well, affinity chromatography is a technique that is used or based on the capacity of the biomolecules or specific non-covalent biomolecules that has the binding capacity to bind to other molecules that we call it as ligands. So what it does, the main principle behind this affinity chromatography is it explores the biological affinity of the macromolecules for its characteristic ligand. For example, you have like hormone receptor binding, enzyme substrate receptor binding, and even antigen antibody binding. So if you see here the structural diagrammatic representation of the ligand affinity chromatography, you have the matrix, the spacer arm, and you have the immobilized ligand. This is the enzyme. What it does, the bound in it just enzyme gets bound to the ligand, so it helps wash free of contamination of contaminating proteins, or non-specific elution, that is pH or ionic concentration change. It just goes affinity elution with the ligand. So what happens is, like, if it is non-specific or non-covalent, it just goes binds there, then restore to optimum conditions, you get the purified enzyme. Or if you see here again, this type, the immobilized ligand plus with the affinity elution on the dialysis process, you get the purified enzyme. Any other ways you can do bind the molecule enzyme with the ligand in order to purify the enzyme. So the types of ligands you see, like substrate analogs, the target molecules are enzyme, antibody antigen, lectin polysaccharide, nucleic acid you have complementary base sequence, hormone receptor, avidin biotin conjugated molecule, calmodulin you have calmodulin binding partner. Then another main thing we have all learned is genetic engineering, glutathione, which is GST fusion protein. Then proteins A and G immunoglobulins. Metal ions, polyhistidine fusion protein. Metal ions, mostly the nickel or cobalt, are being used for polyhistidine fusion protein, the histag. So in the ligand selection, so the ligand that has been selected should interact strongly with the desired macromolecule. However, but it should not interact too strongly. So, and also it should process the functional groups in order to form the covalent linkage with the supporting matrix. So this is what it says in the ligand selection. Now let us see in the ligands attachment is like ligand attachment can be done in two ways. One, activation of the matrix functional groups. Two, covalent attachment of the ligand to these activated groups. So in the activation of polysaccharide, it supports like the polysaccharide supports, like for example agarose involves treatment with cyanogen bromide at an alkaline pH, so pH 11, which is an alkaline stage. So only then activation of polysaccharide supports like agarose when treated with cyanogen bromide only can be activated. If you see here, this is the agarose plus the cyanogen bromide at 11, at the pH of 11. You get iminocarbonate, or if you go like iminocarbonate plus ligand at pH 9, overnight in cold, you get isouria. This is how in one way the activation can take place. So affinity chromatography, it is an exothermic reaction and it needs to maintain temperature constant at 20 degrees centigrade at all times. So there are coupling of amino acid containing ligands to cyanogen bromide activated support is normally carried out by suspending the support and the ligand in a basic buffer solution at the pH of 9. So it is actually gently stirred overnight in a cold room or for 2 hours in the room temperature. Ligand is attached. Following this, the matrix is extensively washed to remove any unbound ligand. So if you see here this is the enzyme, this is the ligand. What it does, what happens is like it goes and binds with that enzyme from the matrix, and then the matrix is being extensively washed. It can be done in two ways, either at at 20-degree temperature overnight when the pH of 9, or it can be done at 2 temperature but gently stirred and continuously for 2 hours, and the matrix is being extensively washed in order to remove any unbound ligand. So if you see the arm, if a ligand is directly attached to an activated group of the support, the macromolecules might encounter the steric restrictions, so might not become adsorbed to the matrix. Some of the chemical metrics are being used as arms are hexamethylene diamine, 3,3-diamino dipropylamine, 1,6-diaminohexane, 6-aminohexanoic acid, 1,4-bis 2,3-epoxy-propoxy-butane, and your spacer arms must possess either any two functional groups. So that's all about the affinity chromatography. Thank you for your wonderful patience in listening to this video. Please like the video, comment, share the video, and don't forget to